Okay, guys, today we're going to talk about enzymes. So we did the catalase lab already in class. We've already done a little video recording about all of that. We've also talked about enzymes your freshman year of biology. So a lot of this is just going to be a quick recap for you. So I've greatly shortened this down from what your textbook actually goes over. So some of these slides are a little bit fouled up, but we're just going to go with it. So this is showing you an enzymatic reaction that uses multiple enzymes to take the initial substrate of A and turn it into the final product of D. So you can see that substrate A is turned into product B by enzyme number 1. Then product B actually turns into the substrate for enzyme number 2, which turns into the product C, and so on. So if you, let's say, had a denaturation event occur for enzyme number 2, what would that do to this overall reaction? If enzyme number 2 has been denatured, what does that mean? Well, it means that the shape has changed and now it doesn't function correctly. Okay, so if you have enzyme 2 that does not work, can you ever make product D? No, because you're still going to have enzyme 1 functioning correctly, right? So you have A turning into B, and A is turning into B, and A is turning into B repeatedly. But B cannot turn into C. So you cannot make product C because enzyme 2 is not working. Well, if you can't have C, then there is no D. So if enzyme 2 is denatured, you're going to have a surplus of product B that's occurring in this overall reaction if that's the enzyme that gets denatured. Okay, think about if enzyme 3 was denatured, you'd have A turning into B, B turning into C, but then C would never turn into D. Okay, so think about that because those are questions that all teachers like to use on their exams, just as a heads up. So we're going to talk about enzymes and how they function and what they are and all of that. So some catabolic pathways are going to be encountered in our discussion of enzymes. So we know that enzymes are protein. You know that they speed up chemical reactions by lower activation energy, and you know that they kick some serious ACE. They end in ASE. Okay, so catabolic pathways are one of the pathways that utilize enzymes, and anabolic pathways would be the other. So catabolic, I like to think cats have claws to tear things apart. So catabolic reactions are going to tear things apart. So here we have catabolic pathways that are going to release energy by doing what? By breaking down our complex molecules into simpler compounds. So we could also say this by taking a polymer, many pieces, and breaking it down to monomers, one piece, something big, and breaking it down. Okay, and cellular respiration is a great example of this because you're going to take glucose and in the presence of oxygen, you're going to break it down and to eventually help us create ATP through the final stages of the electron transport chain and all of that when we're doing cellular respiration. On the flip side, like I said, anabolic pathways are actually going to be building. Think about like anabolic steroids. I like to pick things up and put things down. We're talking about like growing your muscles. Okay, in this case, we just have anabolic pathways that are going to consume energy in order to build complex molecules. So this would be like taking our monomers and putting them together to make a polymer. Again, a couple of single pieces added together to make one larger piece. Okay, so the synthesis of protein from amino acids is an example of anabolism because you have a protein, a polymer, being made from a whole bunch of tiny monomers. Also, my dogs are in the background going psycho. I apologize for their skidding. So how organisms kind of balance this consumption of energy and this expulsion of energy through anabolic and catabolic pathways, respectively, is called bioenergetics. So this is a study of how organisms are going to manage their energy resources. And there's a couple different forms that this energy can exist in. So energy as a whole is just the capacity to cause change. Like I said, dogs in the background being crackheads, I'm sorry. Okay, so energy exists in a couple different forms that can perform work. So a cell does 3 main kinds of work, and all of this is going to be powered by ATP. Okay, so ATP powers cellular work by coupling our exergonic reactions, or ones that release heat or energy, to our endergonic reactions, which are reactions that actually consume heat or energy. So the cell does 3 main kinds of work, like chemical work, transport work, which we've talked about previously in cellular transport, and mechanical work. So in order to do this work, we need energy. Okay, energy sometimes called heat when you're reading about like thermodynamics and everything during this chapter. Okay, but to do work, cells have to manage their energy resources by coupling these exergonic processes to help drive the endergonic processes. And again, exergonic is when we are releasing energy or heat from a reaction, and endergonic is when we need an input of energy or heat into a reaction. So they're coupling them together. Think about like redox reactions, like reduction and oxidation reactions. They're coupled always. That's how these work as well. So in order to manage all of this energy, we have to couple the exergonic processes to help drive the endergonic processes. Okay, and most of this coupling is aided through ATP. So like ATP is our cellular currency of energy. It is the big boss that helps us do all of this stuff that requires energy. And we're going to talk about the structure of ATP and how that's helpful in just a few minutes. So ATP, adenosine triphosphate, again, triphosphate, 3 phosphates, okay, is made of a ribose sugar, an adenine, which is a nitrogenous base, and then 3 phosphate groups. Now, it's important it has 3 phosphate groups. That's why it is triphosphate, okay? So, again, this is our energy source. So how do we use it? We have to put it through a hydrolytic process, so hydrolysis, hydro-water lysis to break. Again, hydro-water lysis to break. We're going to put it through a hydrolysis reaction in order to break the ATP to release its energy. So here you can see that you have an ATP up at the top. You have your ribose right here, you have your adenine, and then you have your 1, 2, 3 phosphate groups. This is the structure of ATP. Okay, so when we take ATP, this is the same picture, just a little bit simplified. Okay, ATP, we add water, hydrolysis, to break with the addition of water. We're going to break a terminal phosphate off. So right here, this is the bond that we're breaking in order to release inorganic phosphate. And all that means is that it doesn't contain carbon to hydrogen bonds. True. Okay, and then we have ADP, adenosine diphosphate, and then a release of energy. So this reaction would be used to drive chemical reactions. So like I just said, ATP, adenosine triphosphate, can be broken down to adenosine diphosphate with the release of an inorganic phosphate when we put it through hydrolysis. So again, hydro-water lysis to break. So when we add in water, it releases a terminal phosphate off of ATP, creating ADP, and this expels energy. So energy that's released from ATP when the terminal phosphate is broken helps to drive chemical reactions. So this release of energy comes from the chemical change to a lower energy state, okay? And it's not actually coming from the phosphate bond itself. Having all those phosphates together, because they're highly negatively charged, like it's building up a whole lot of like pressure essentially. Releasing one of them lowers the energy state, which is going to help to drive the reaction. So like I said earlier, there's the 3 different types of cellular work that our bodies, our cells do. Mechanical work, transport work, which again we've talked about, and chemical. Chemical is something we're going to be focusing a lot in chapter 7 and 8. Okay, so these are all powered by the hydrolysis of ATP, which we just looked at. Okay, so in a cell, the energy from the exergonic reactions, or the reactions that help to release energy of ATP hydrolysis, are then used, it's like harnessed and then used to drive our endergonic reactions, because you have to have an input of energy. And if you're releasing energy from ATP, then you have a surplus of free energy that you can then put into these endergonic reactions to help drive them forward. So again, all of these are coupled reactions. You have endergonic and exergonic kind of working together. Think about it again like redox reactions. Reduction and oxidation have to occur together. These are the same way. So ATP drives endergonic reactions, the endergonic, so input of energy, by phosphorylation. So it's transferring a phosphate group to some other molecule, usually a reactant. ATP with 3 phosphates donates a phosphate to phosphorylate something else that does not necessarily have a phosphate. Okay, so the recipient, typically the reactant, okay, is called the phosphorylated intermediate. Well, this makes sense because it's been changed and it is not yet a product. It is an intermediate. And it is phosphorylated because we have just slapped a phosphate group onto it. So that's aptly named. Okay. ATP hydrolysis leads to a change in a protein's shape and often its ability to bind to another molecule. So if you have ATP hydrolysis that helps to release a phosphate group, and that phosphate group attaches to a protein, it typically alters something about it, whether it's activating it or deactivating it. So you can phosphorylate or dephosphorylate various substances, and that controls what that substance does. It all has to go back to that structure dictates function sort of thing. So when we're adding a big negative phosphate group onto something, you're changing its overall structure, which means that you're temporarily changing its function. So keep that in mind. Here's some examples with like transport work here. So we have ATP that's phosphorylating a transport protein. So you see ATP, it's donating a phosphate to the little purple protein there, which causes a conformation change. So the protein opens up to the opposite side of the cellular membrane. And of course, because you have released the phosphate there, you have ADP, right? Because you've just removed a phosphate from it. You also have an example here in mechanical work. So these are like the little dynein, like little motor proteins that are helping to move things around the cytoskeleton. So ATP binds non-covalently to motor proteins and then is hydrolyzed. Okay, so you see ATP that binds and then you don't have any motion happening yet, right? But then you see motion actually occurring once it's hydrolyzed. So once you have that release of the terminal phosphate, you're releasing some of that pressure of all the phosphates pushing on each other to a lower energy state, which is going to help to drive the cytoskeletal transport system here with this motor protein. So luckily for us, ATP is a renewable source, right? It's not like our fossil fuels that like once they're gone, we're SOL, right? Completely screwed, right? So ATP is renewable because it's just ATP. If you have ADP, which is the product of all these reactions, you just put another phosphate on it and suddenly you have 3 again. It's called ATP and it's ready to go. Okay, so ATP is a renewable resource that can be regenerated by the addition of a phosphate group to adenosine diphosphate. So like I said, at the end of all these reactions, you have this guy called ADP. Well, guess what? If there's a random phosphate floating around in the cytoplasm or something, you can just grab onto that and attach it back onto our ADP to create ATP. So if you break something, you can typically put it back together, and that's kind of nice in these specific reactions. So the cell is going to use this energy to phosphorylate ADP. It's coming from our catabolic reactions. Okay, so catabolic reactions, again, break things down. So like a prime example is going to be cellular respiration, which we're going to get into pretty soon in the next chapter or two. Okay, so cellular respiration takes glucose and breaks it down into these little tiny particles of it, okay? And then we're going to use that to help to generate ATP. Where does this happen? The mitochondria, because it's the powerhouse of the cell. If you know anything about biology ever in your life, it's that the mitochondria is the powerhouse of the cell. Okay, so this is where these reactions are occurring in order to help generate ATP. Well, how do we generate ATP? By taking an ADP and putting an extra phosphate on it to make 3 phosphates to make ATP. Okay, so the ATP cycle is like a revolving door. So we have energy that's going to pass from our catabolic to anabolic pathways and back again because we're constantly breaking and remaking our ATP. So it's this cyclic thing that we see going on here. So you have energy from catabolism, which is when we have exergonic, a release of energy that's going to help drive our ATP turning back into our ADP, right? We have this constant exergonic endergonic coupling that's occurring here. You have your ATP meeting up with a water molecule through hydrolysis that's going to break it down into ADP, an inorganic phosphate. That phosphate can phosphorylate something to change the shape of it to either activate or inhibit its function, such as other proteins and enzymes, right? And then we can always just go back and regenerate all of this energy by just reintroducing that terminal phosphate group back onto our ATP molecules. That way we have our cellular currency ready to go for the next reaction that requires energy. Okay, so enzymes, we've been talking about. Enzymes speed up metabolic reactions by lowering energy barriers. Now, how are we used to saying this? Enzymes speed up chemical reactions by decreasing activation energy. That's what it does. So an enzyme is a catalyst, and this is some sort of chemical agent that's going to help to speed up a chemical reaction without being consumed by the reaction. So let me just say that one time again for those of us that can't remember anything and for those of us that need to wake up and listen. Okay? A catalyst speeds up a chemical reaction, but it is not consumed in the reaction. So you have an enzyme. It's kind of like a lock and a key, like with its substrate. One enzyme functions for one substrate, and that's kind of it. Think about a key. Your house key opens up your house. Your house key will not turn on your best friend's dad's car. Why? Because it's not meant for that. It doesn't fit right. It's just wrong. Okay, so you have a certain lock and a certain key, and they are made for each other, and they are a happy little couple, and that's how it works. Okay, so a catalyst is specific for its reaction, but it's not used up in the reaction. Think about when you go home every day, or if you're old enough to drive, you're driving to or from school every day. You put your key in the ignition, you turn it, you drive home, great, awesome. Take your key out in the morning, you go back to school, awesome. You start your car up again. It's not like you need a brand new key every single time you get in your car to drive, or every single time you go home, you don't need to unlock it with a brand new key, right? You just use the same one again and again and again. Now, of course, if your key breaks, well, sure, then you need a new one because it's broken, hello, right? But enzymes function like a key. They're meant for one specific function to open that lock or to catalyze this specific reaction, and you keep using them over and over and over and over and over again until something happens to it, okay? So that's really important to remember about our enzymes. They are still functional even after they have completed. So here we have an enzymatic reaction that we're going to look at. So this is breaking down sucrose into glucose and fructose. This is actually got to be a 12 here because it's a 1 to 2 to 1 ratio because OSE means it is a carbohydrate. So sucrose. Okay, we add water and also something called sucrase. ACE means that it's going to kick some ACE. It is an enzyme and is going to help speed up this chemical reaction. So we have a dimer, so two pieces that we're breaking into monomers, glucose and fructose. So our enzymes function by lowering the activation energy. Okay, so every chemical reaction between molecules involves bond breaking and bond formation in order to generate our products and break down our reactants. So the initial energy that's needed to start a chemical reaction is called our activation energy or energy of activation. Okay, so this is how much energy is needed to activate the chemical reaction. Okay, so the activation energy is supplied in the form of thermal energy, so some sort of heat typically, that the reactant molecules absorb from the surroundings. So if we can limit that, if we can lower that, it's not like the enzyme, the reaction doesn't have to wait around very long for all of these reactants to absorb energy in order to get to a higher energy state and then start the reaction, okay? So with our enzymes, we're lowering the activation energy, which is why it happens faster because our reactants aren't just like chilling around trying to wait for some energy to be transferred to them in order to proceed to make a product. So here's a typical reaction. You have a reactant that goes through a transition state, like an intermediate, and then you make a product. So look at the, it's kind of covered up by the little black arrow here, but it's like the energy of activation here. You're waiting for these reactants to absorb enough heat, thermal energy, in order to reach a transition state before they can come back down and free energy to the product. Now look at it with an enzyme, okay? So the enzyme is this red line. And this is actually happening a whole lot faster. But look, you're starting off at the same energy because the reactants are not different. And you're ending at the same energy because the products are not different. What's different is right here. It's the energy of activation. Okay, and this is different because you're not having to wait. You're using the enzyme to help you overcome that buildup of energy. So you can just use the enzyme to make this reaction happen a lot faster. Okay, so notice again, you start at the same energy state and you end at the same energy state. What's different is the energy of activation. So the black line is without the enzyme. You need a lot more activation energy built up before the reaction will actually proceed. Whereas with the enzyme, it's a lot less energy that is utilized because you have the addition of the enzyme. So like I said earlier, our enzymes are like locks and keys, okay? So they work on one specific substrate. So the reactant for an enzyme is called a substrate. So the enzyme functions on one specific substrate. So the reactant is the substrate. They're the same thing, okay? Then when the enzyme is bound to the substrate, it forms a complex called the enzyme substrate complex. Really creative name there. It just means enzyme is attached to the substrate. Congratulations. How are they attached? They're attached through something called the active site. The active site is the region on the enzyme where the substrate physically binds and attaches to the enzyme, okay? Then the enzyme has like a complementary fit. It's like a change in shape that can happen, like an induced fit model, which we'll look at, between the shape of its active site and the shape of the substrate. So they fit together exactly like a lock and key. And sometimes in an induced fit, we have the enzyme actually change shape to actually like bond tighter to the substrate itself. Okay, so our enzymes change shape due to the chemical interactions with the substrate. So like I just said, the induced fit is when we have the enzyme that's going to bring in the substrate and chemically bind to it and change shape. So let's look at that in a picture here. Again, my dogs are being annoying. I apologize. So the enzyme is this big blue guy here. This is what the molecular view of that would look like. Then we have the active site right here, which is where our substrate is going to bind. But you can see the substrate is very small and the active site is kind of huge. So then when we have the substrate actually bond to our enzyme, you can see that this is an induced fit because we have the enzyme actually wrap around the substrate complex here. Okay, so the catalytic activity of an enzyme actually occurs in the active site. Okay, so this is where all of the reaction actually occurs. So we have the substrate that binds to the active site of the enzyme. The active site can actually lower the energy of activation by doing one of these things, by helping to correctly orient the substrates, by straining the substrate bonds, by providing a favorable microenvironment for the reaction to occur, or covalently bonding to the substrate. So here's an example of how this kind of works overall. Yes, I know that the circles are not in the correct places and that they don't have numbers on them. I mean, it is what it is sometimes. So we're going to start up at the top. Substrates are going to enter into the active site. So our substrates, also called reactants, are going to bind to our enzyme in the active site. So the substrates are held in the active site by weak interactions. Okay, then here we have the enzyme substrate complex. It's going to go through a reaction in order to make our products. So the substrates are converted into our products. Now, products are released from the enzyme. Now, remember, the enzyme is not utilized. It's not used up completely. Okay, we just use it again. Just like your house, you can just keep using the same key to get home every day. Okay, so now the active site is available for new substrates to bind. And this will just keep happening again in this big cycle, this big circle, until something happens to either just the active site or this enzyme as a whole in order to prevent it from correctly bonding to its substrates. It will keep working until the conditions change and cause it to stop functioning correctly. Okay, so an enzyme's activity can be affected by things like environmental factors like temperature and pH. So those are going to be your biggest ones. Okay, so if you have temperature that's not optimal or pH that's not optimal, kind of like we saw in the catalase lab that we did recently, your enzyme is not going to function correctly. It might function a little bit, but its optimal temperature and pH are where it's going to have the highest enzymatic reactivity. So the enzyme has an optimal temperature, like I said, that it functions at an optimal pH. So if you think about like us as humans, our body temperature is what? It's 98.6 degrees Fahrenheit or about 37 degrees Celsius. So when we get fevers that kind of get out of control, it starts to get dangerous because then some of your enzymes could potentially stop working. Same thing as your body cools down too much, okay? So like in our catalase lab, like our enzymes worked in the warmer temperatures because if you think about it, you know, I had it in the freezer. It shouldn't work in the freezer because the animal that I got it from, which is a cow, is not frozen when it's alive, right? It's a warm-blooded animal. So when it's alive, its body temperature is going to be greater than 0 degrees Celsius, which means for its enzymes to be optimally functioning, it better be hotter than 0 degrees Celsius. So here's some examples of that. We have the optimal temperature for the typical human enzyme, which I said was about 37 degrees Celsius. Does that mean it doesn't work at all anywhere in here? No, it does work. It's just optimal at the peak, which is at 37 degrees. According to this, which that looks like more than 30. It's probably messed up a little bit, kind of like all those circles and everything. You know, we got to forgive these textbook writers sometimes. All right. And then the optimal temperature for a thermophilic or heat tolerant bacteria, you can assume is going to be a heck of a lot higher than what a human enzyme would be, because we are not thermophilic and heat tolerant. Hello, Texas. Here's looking at you, right? We do not function at the same temperatures. Therefore, our enzymes are not going to function at the same temperatures. And we see the same thing down in the bottom graph, but talking about pH. Of course, our pH for pepsin, which is an enzyme that functions in our stomachs, okay, is going to be different than it would be for trypsin, which is an intestinal enzyme. The stomach has a much higher acidity rating than the intestines, right? So you wouldn't expect that the same pH would be the optimal pH for the enzymes that are found in these different locations throughout the body. We also have something called cofactors and coenzymes. So enzymes don't always function by themselves. Sometimes they need a little bit of help. So we have things that are called cofactors, which are non-protein enzyme helpers. A cofactor is typically inorganic, which means it does not contain carbon to hydrogen bonds. Typically, it's a metal. The most common one in humans is like calcium. Okay, they can be organic as well, which would mean it contains carbon to hydrogen bonds. If we have an organic cofactor, it's typically called a coenzyme. And our coenzymes as humans typically include vitamins, and there's a whole array of different vitamins that are utilized as coenzymes in the body. We also have inhibitors. So we've seen how we have our reactants, we have products, we have the induced fit, things like that. Now we have something called inhibitors. Now inhibitors, the word inhibit means to prevent or to stop. So competitive inhibitors bind to the active site in order to stop the substrate from binding to the active site. So it's in competition with the substrate in order to bind, to the active site. Okay, so then we also have non-competitive inhibitors. So this is not going to bind to the active site. It's not competing with the substrate. It's actually going to bind to a different site of the enzyme, and it's going to change the whole shape of the enzyme, making it less likely to fit or to match up with the substrate. Okay, so some of these examples of inhibitors could be toxins, poisons, pesticides, and antibiotics. The latter is something that, we put in our bodies voluntarily, but typically our antibiotics would inhibit enzymes in like bacteria and things like that, whereas like toxins, poisons, pesticides, and things like that are going to be more dangerous for us because they could inhibit our own enzymes as well. So here's an example of what a normal binding situation would be for a substrate and an enzyme through the active site. A competitive inhibition reaction here, you have the enzyme with a competitive inhibitor. Now, this is competitive because it is competing for the active sites. My dogs are being crackheads. Can you stop? You have the inhibitor binding to the active site. It is literally competing for that spot. It's competing with the substrates. Okay, and then in non-competitive inhibition, we have he's a little sneaky guy, right? He's down at the bottom, and he is not competing with our substrate, but he is binding to the enzyme and causing a conformation change. Look at this, looks like a little thumb sticking up. It's now sticking out to the side, right? So this non-competitive inhibitor has bound to the enzyme and has completely fouled up the shape here. So now our little substrate will not bind correctly. So it's still inhibiting the reaction from occurring because it's changing the shape of the enzyme. Here, our competitive inhibitor is literally just taking up the space where the substrate should go. Again, it is stopping the reaction. Can these both come off later? Yes, absolutely. But just in this picture, these reactions would be inhibited because there's an inhibitor. Okay, so enzymes are proteins that are encoded by our genes, and we have gene regulation, right? That's going to help us decide when we need certain enzymes to be utilized. We can turn them on and off. Okay, so changes and mutations in our genes lead to changes in amino acid composition of our enzymes. And if we have altered amino acids in our enzymes, it can alter the substrate specificity, which means that maybe the substrate doesn't bind correctly to the enzyme anymore. Maybe it catalyzes a different reaction. So under new environmental conditions, a new enzyme might be more favorable. So this can kind of like be a source of evolution, right? If you have a desirable trait, it's going to be perpetuated in the population over millions of years. So when we have small environmental changes and we have small mutations that occur in our enzyme structure function, right, then this can lead to evolution. Okay, so chemical chaos would result if a cell's metabolic pathways were not tightly regulated. So regulation of these enzymes, because it's encoded on our genes, which is DNA, right? Remember, we talked about protein synthesis. So when you want to just code for a specific protein, an enzyme is a protein, you're only dealing with specific genes, right? So our genes are evolving, but our genes are part of the DNA, and you can actually regulate when you make certain proteins or enzymes, because they're the same thing, right? So regulation is something you just need to be aware of, but you don't need to know all of the specifics anymore, and you're welcome for that, okay? But essentially, the cell can switch on or off specific genes that it needs to accomplish specific tasks, which makes sense. It is a tightly regulated and controlled array of reactions that's happening. We have allosteric regulation as one type of regulation that occurs. So it can either inhibit or stimulate an enzyme's activity. So essentially, an allosteric regulator is actually affecting the enzyme. Hello? Rude. Not the genes. Okay, so it's actually affecting the enzyme. Once we've already created the protein, it's already folded, it's already a functional enzyme. This is when we're going to regulate this particular type of regulation. Allosteric regulation is once we already have the functional enzyme. Okay, so allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. So essentially, it kind of works like the inhibitor, the non-competitive inhibitor. You have an allosteric regulator that's going to bind somewhere, and it will either turn on or off the enzyme. Okay, so we're just going to look at what that actually looks like, because all of this is saying the same thing as this picture. So you have allosteric activators and inhibitors. So here you have the allosteric enzyme with 4 subunits. You have the active site 1, 2, 3, 4, and you also have this regulatory site 1 of 4, 1, 2, 3, 4. You have an activator, which is going to turn it on or activate it, okay, that's going to bind, and this is a stabilized active form. Now, if you have a non-functional active site here, it's probably because you have an inhibitor that is bound, okay? This is a stabilized inactive form. So in this regulatory site, we have an inhibitor, which is going to prevent the reactions from occurring. Whereas in the regulatory site, if we have an activator, this will promote the reaction, okay? And then we also have a cooperativity reaction here. So it's another type of allosteric activation. This is when we have a substrate that's going to bind to form the stabilized active form. So we actually have to have the binding of a substrate in order to like turn on the enzyme, because you see all these other sites, it can actually go and attach to other things in order to catalyze multiple reactions kind of at the same time. So cooperativity is a form of allosteric regulation that can amplify enzyme activity. So like I said, if you have the binding of a substrate that's going to turn it on, well, then it will also want to bind the other 3 substrates. Okay, and of course the substrates are typically the same. You can see in this picture very clearly that they would be the same, right? So we have one substrate molecule that primes the enzyme. So then it's going to want to act and add on additional substrates more readily. than it would before, because look at the shape change, right? So the substrate binding actually activates this. It's also binding to a substrate on one of the active sites, which can affect the catalysis on different active sites, right? So these are all wanting to complete the same catalytic reaction, or catalyze the same reaction that's happening here. Not necessarily catalytic. Okay, so they're all going to now have a greater affinity for the substrate in order to bind to the substrate, and then all of the reactions will ensue at the same time. So we have structures within the cell to help bring order to our metabolic pathways. Like I said earlier, we have like the mitochondria, the powerhouse of the cell, is going to help us create our ATP and things like that. So we have like our cellular respiration enzymes that are going to be located in the mitochondria, and you have your hydrolytic enzymes that are located in lysosomes of our eukaryotic cells, specifically our animal cells, right? So we have specific localizations of enzymes within our cell based on what their function is, right? Because again, structure dictates function. So we have the different shapes of the different enzymes that are going to help to localize them in different areas that are going to be the most advantageous when they're going to come into contact with their substrates the most often, right? So like if you have all these enzymes for cellular respiration located in the mitochondria, well, all of that's going to be close together because that's where all of the substrates and products need to be localized in order to drive the overall reaction called cellular respiration in order to make our ATP, right? Lastly, one more time, this is the overall graph of what our enzymes do. Okay, so enzymes speed up your chemical reactions by lowering the activation energy. That's what this red line is representing. Again, the energy state of the reactants and the products do not change. What actually changes is just the energy that is input, because you have the enzyme here, and here you are waiting for the thermal energy to be absorbed by the reactants, which takes forever. Okay, so the addition of our enzyme is going to speed up this chemical reaction. Thank you. Have a great day.